Hello, my name is Noriko Kamei from the University of California, Irvine in the United States. Thank you to the organizer for giving me the opportunity to present my work. Today, I would like to talk about our recent accomplishment. The running title is "The Injured Inflammatory Environment Overrides the Epigenetic Landscape Shaped by TET2." Preclinical animal studies suggest that neural stem cell transplantation is a promising therapeutic strategy for spinal cord injury. However, the environments following spinal cord injury exhibit prolonged multiphasic cellular inflammatory responses as shown in the figure. The acute phase is characterized mainly by polymorphonuclear leukocytes, whereas macrophages characterize the subacute to chronic phases. Transplantation therapy and transplanted cell performance in the injured spinal cord will remain important for predicting how cell lines perform in the transplantation microenvironment. However, how different microenvironments influence their epigenetic states remains poorly understood. Therefore, we created several pluripotent stem cell-derived human neural stem cell lines using the same protocol under xeno-free conditions. Then compare changes in the expression of enzyme regulating DNA methylation, DNA methyltransferase genes, DNMT1, DNMT3A, and DNMT3B, and demethylation, methylcytosine deoxygenase 1011 translocation genes, TET1, TET2, and TET3 in these cell lines. As shown here, TET2 gene expression is significantly upregulated in human neural stem cells derived from pluripotent stem cells compared with pluripotent stem cell itself. TET2 expression levels vary across cell lines, even though we generate each human neural stem cell using the same protocol. This variation might be due to intrinsic factors. In this study, we used Chef6 embryonic stem cells to generate unsorted ES-derived neural stem cells and sorted ES-derived neural stem cell by magnetic activated cell sorting with positive selection for the cell surface stem cell marker CD133. We also sequentially eliminated CD34, a hematopoietic stem cell marker. To minimize the potential contribution of variation observed between parent lines, we focus on these cell line derivatives, all of the same ES origin as shown here. These derivative undifferentiated ES-derived neural stem cell lines show comparable levels of TET2 expression. We differentiate among these ES-derived neural stem cell lines shown on the previous slide. TET2 expression levels increase to varying degrees. The red bar for hES-NSC shows glial propensity, whereas the blue bars, especially the dark blue bar, shows neuronal propensity. These data suggest a relationship between TET2 upregulation levels and neuronal lineage propensity. For future experiment and comparisons, we will refer to the glial propensity cell line as glial M7 and the neuronal propensity cell line shown in the dark blue bar as Neuro M28. Also, we noticed that even at the undifferentiated stage, these cells already show specific messenger RNA expression of selected neural markers. These cells could be primed toward a specific cell fate. To understand TET2 role in these cell lines, we manipulate the intrinsic epigenetic status by knocking down the TET2 gene or the DNMT3A gene. I did not go into detail about these data, but they show successful gene knockdown results. However, TET2 knockdown was ineffective in undifferentiated Neuro-M28. After successful knockdown for each gene in each cell line, we split these cells into two differentiation media, normal differentiation media DM and macrophage condition media, which mimic subacute to chronic inflammation in the injured spinal cord, were used to differentiate them for 14 days. Surprisingly, the same knockdown undifferentiated cells alter cell differentiation and responses as analyzing using the selected marker gene under the normal versus inflammatory conditions. These data suggest that the inflammatory condition can override or modify epigenetic regulation in these cells. Next, we wanted to know what kind of epigenetic landscape is associated with glial or neuronal preference in each neural stem cell line. Therefore, we analyzed genome-wide differential methylation and hydroxymethylation in these undifferentiated glial M7 and Neuro M28 using reduced representation by sulfide sequencing RRBS, which detect methylcytosine, reduced representation hydroxymethylation profiling RRHP, which detects hydroxymethylcytosine, and RNA-seq as shown. The results showed that the combination of RNA, RRBS, and RRHP at the undifferentiated stage detects many genes and transcription factors. We focused on transcription factor genes because they are key regulator of cell fate and differentiation. 70% of RRBS and RRHP detected as significant in the genome in undifferentiated glial M7 and Neuro M28 did not overlap with gene expression results. Particularly, many genes and transcription factor are detected only by RRHP. And these genes and transcription factor are also involved in neurogenesis. Typically, hydroxymethylcytosine is enriched in gene bodies and an active or poised enhancer on the genome and is involved in gene expression. These undifferentiated glial M7 and Neuro M28 cells could be primed for their respective fates prior to differentiation. Next, we tracked the methylation and hydroxymethylation patterns detected at the undifferentiated stage alongside each cell line's differentiation. The top graphs show the number of transcription factor detections under normal differentiation DM and macrophage condition media for glial M7 and Neuro M28. Exposure to the different differentiation environments affected the overall number of transcription factors detected with 5MC, 5HMC marks. Some transcription factor genes were detected at the undifferentiated stage, which we call trackable, whereas others were newly detected at the differentiated stage, which we call untrackable, anti. 90% of transcription factor are shared between normal and macrophage condition media as shown in each Venn diagram. Additionally, 20 to 30% of these overlapping transcription factors enabled us to detect a switch in the 5-HMC pattern between normal and inflammatory conditions. In the meantime, TET2 expression levels in these cell lines are shown at the bottom. MAC condition media reduce TET2 gene expression, which correlate with fewer transcription factor genes detected with 5-HMC on the top graphs. We asked the next question: does MAC condition media mimics the in vivo microenvironment of the subacutely to chronically injured spinal cord? We used the in vivo paradigm shown here. We used immunodeficient mice to create a T9 spinal cord injury. We then transplanted either glial M7 or Neuro M28 at 9-day post-injury. We recovered engrafted cells from the injured animal spinal cord 28 days post-transplantation. Then we could isolate RNA and DNA from engrafted cells. We analyzed RNA for RNA-seq and DNA for RRBS to detect methylation. However, we failed to obtain a sufficient number of mappable CPG sites to generate reliable data. So the following data are from RNA-seq results. Principal component results for engrafted glial M7 and Neuro M28 are shown on the left. 7 biological replicates per cell line were analyzed. The results for differentially expressed genes are shown on the right. 199 genes, including 1 transcription factor, are upregulated in glial M7, showing red, but downregulated in Neuro M28. 3,478, including 369 transcription factor genes, are upregulated in Neuro M28, shown blue, but decreased expression in glial M7. The left panel shows the volcano plot of differentially expressed gene between glial M7 in red and Neuro M28 in blue. Interestingly, in an in vivo injured environment, TET2 gene expression was significantly upregulated in engrafted Neuro M28 compared with engrafted glial M7. Among the 199 differentially expressed genes upregulated in engrafted glial M7, the represented genes were mainly involved in extracellular matrix and immune responses, including hallmark genes of epithelial mesenchymal transition. By comparison, 3,478 differentially expressed genes are upregulated in engrafted Neuro M28. 369 transcription factor genes are associated with the neuronal pathway. In particular, DCX and MAP2 genes are highly expressed. 62% of transcription factor from engrafted Neuro M28 were also detected during in vitro differentiation, which we could track, indicating a strong overlap with in vivo. However, unexpectedly, more than half of these transcription factor genes detected in vitro are observed only under normal differentiation condition. Complex inflammatory environment of the injured spinal cord may influence TET2 expression, although further studies are needed to understand this process. From this study, upregulation of TET2 is a potential pluripotent exit signal that regulates the epigenetic landscape in human pluripotent stem cell-derived neural stem cells. TET2 plays a major role in shaping the epigenome as embryonic stem cell transition into neural stem cells. TET2 expression is critical for guiding cell fate and regulating differentiation under the normal differentiation. However, the inflammation overrides the cell's primed TET2 activity, effectively altering the cell's ultimate differentiation fate. Take-home messages are: tracking 5MC 5HMC changes allowed us to detect a switch in 5MC 5HMC patterns between the normal and inflammatory conditions, which may result from TET2-associated methylation dynamics. TET2 expression levels and detection of epigenetic marks 5MC 5HMC could be informative to predict changes that occurs in different environments prior to gene expression. The inflammatory niche at an injured site actively changes its epigenetic networks, a fact that must be accounted for in future cell transplantation therapies. This work was supported by the Wings for Life Spinal Cord Research Foundation grant. I would also like to thank Dr. Anderson and Dr. Cummings at University of California, Irvine for their support of this study, Dr. Luo at UC Los Angeles for assistance and suggestions. I also thank the Genomic Research and Technology Hub at UC Irvine. Thank you very much for your attention.